agriculture has progressed dramatically since early 1970s as a result of research and development based policy support. High yielding varieties and hybrids along with intensive inputs, particularly the chemical fertilizers, pesticides and water, have resulted in a phenomenal growth. Developing countries, being largely agriculture based economies, benefited substantially from these advancements, achieving food self-sufficiency, improved rural incomes and higher overall economic growth. While the gains have been very impressive, the input intensive agriculture has resulted in some undesirable effects on the environment and the overall sustainability of the farming systems. Indiscriminate use of fertilizers, particularly the nitrogenous ones, has led to substantial pollution of soil, air and water. Fertilizer contamination of groundwaters has led to eutrophication of lake and river waters, causing depletion of oxygen and even death of aquatic life, nitrate pollution, increased emission of gaseous nitrogen and metal toxicities. The presence of nitrates in potable water has been blamed for health hazards such as birth defects, impaired nervous system, cancer and the blue baby syndrome. Very often microorganisms are not as efficient in natural surroundings as one would expect them to be, and therefore, artificially multiplied cultures of efficient selected microorganisms play a vital role in accelerating the microbial processes in soil. Biofertilizers are microbial inoculants supported on carriers and make the agriculture environmentally sustainable, economically rewarding and intellectually stimulating. These are microbial inoculants that are carrier based preparations containing beneficial microorganisms in viable state. They are intended for seed or soil application and designed to improve nitrogen and potassium of soil and provide plant growth promoter substances. Use of biofertilizers is one of the important components of integrated nutrient management, as they are cost effective and renewable source of plant nutrients to supplement the chemical fertilizers for sustainable agriculture. Several microorganisms and their association with crop plants are being exploited in the production of biofertilizers. They can be grouped in different ways based on their nature and function. Microbial inoculants are artificially cultured strains of certain soil organisms that can improve soil fertility and productivity. Rhizobia was first discovered in 1895, followed by Azotobacter and then blue-green algae and a host of other microorganisms. Azospirillum and vesicular arbuscular mycorrhizae, that is VAM, are fairly recent discoveries. Biofertilizer, when applied to seeds, plant surfaces or soil, colonizes the rhizosphere or the interior of the plant and promotes growth by increasing the supply or availability of primary nutrients to the host plant. Biofertilizers add nutrients through the natural process of nitrogen fixation, solubilizing phosphorus and stimulating plant growth through the synthesis of plant growth promoting substance. Biofertilizers can be expected to reduce the use of chemical fertilizers and pesticides. The microorganisms in biofertilizers restore the soil's natural nutrient cycle and build soil organic matter. Through the use of biofertilizers, healthy plants can be grown while enhancing the sustainability and the health of the soil. Since they play several roles, a preferred scientific term for such beneficial bacteria is plant growth promoting rhizobacteria, that is PGPR. They are extremely advantageous in enriching soil fertility and fulfilling plant nutrient requirements by supplying the organic nutrients through microorganisms and their byproducts. Hence, biofertilizers do not contain any chemicals which are harmful to the living soil. Biofertilizers provide eco-friendly organic agro input and are most cost effective as compared to chemical fertilizers. Biofertilizers such as Rhizobium, Azotobacter, Azospirillum and blue-green algae, also called as BGA, have been in use for a long time. Rhizobium inoculant is used for leguminous crops. Azotobacter can be used with crops like wheat, maize, mustard, cotton, potato and other vegetable crops. Azospirillum inoculations are recommended mainly for sorghum, millets, maize, sugarcane and wheat. Blue-green algae belonging to a general cyanobacteria genus Nostoc or Anabaena or Tolipothrix or Olocyra fix atmospheric nitrogen and are used as inoculations for paddy crops grown both under upland and lowland conditions. Anabaena, in association with water fern Azolla, contributes nitrogen up to 60 kg per hectare per season and also enriches soils with organic matter. Other types of bacteria, so-called phosphate solubilizing bacteria such as Pentoia agglomerans strain P5 or Pseudomonas putida strain P13 are able to solubilize the insoluble phosphate from organic and inorganic phosphate sources. In fact, due to immobilization of phosphate by mineral ions such as iron, aluminum and calcium or organic acids, the rate of available phosphate in soil is well below the plant's need. In addition, chemical inorganic fertilizers are also immobilized in the soil immediately, so that less than 20% of added fertilizer is absorbed by plants. Therefore, reduction in inorganic phosphate resources on one hand and environmental pollutions resulting from both production and applications of chemical phosphate fertilizer on the other hand have already demanded the use of new generation of phosphate fertilizers globally known as phosphate solubilizing bacteria or phosphate biofertilizers. Rhizobium. Rhizobium is a soil habitat bacterium which is able to colonize the legume roots and fixes the atmospheric nitrogen symbiotically. The morphology and physiology of Rhizobium will vary from free living condition to the bacteroid of nodules. They are the most efficient biofertilizer as per the quantity of nitrogen fixed is concerned. They have 7 genera and highly specific to form nodule in legumes, referred as cross inoculation group. Azotobacter. Of the several species of Azotobacter, A. crocococcum happens to be the dominant inhabitant in arable soils capable of fixing nitrogen. 2 to 15 milligrams of nitrogen is fixed per gram of carbon source in culture media. The bacterium produces abundant slime which helps in soil aggregation. Azospirillum. Azospirillum lipoferum and Azospirillum brasiliensis are primary inhabitants of soil, the rhizosphere and the intercellular spaces of root cortex of gramineous plants. They perform the associative symbiotic relation with the gramineous plants. Apart from nitrogen fixation, growth promoting substance production, for example, indole acetic acid IAA, disease resistance and drought tolerance are some of the additional benefits due to Azospirillum inoculation. Cyanobacteria. Both free living as well as symbiotic cyanobacteria, that is blue green algae BGA, have been harnessed in rice cultivation in India. A composite culture of BGA having heterocystis nostoc, anabaena, olosyra etc. is given as primary inoculum in trays, polythene lined pots and later mass multiplied in the field for application as soil based flakes to the rice growing field at the rate of 10 kg per hectare. Azolla. Azolla is a free floating water fern that floats in water and fixes atmospheric nitrogen in association with nitrogen fixing blue green algae Anabaena azolei. Azolla is used as biofertilizer for wetland rice and it is known to contribute 40 to 60 kg nitrogen per hectare per rice crop. Phosphate solubilizing microorganisms or PSMs. Several soil bacteria and fungi, notably species of Pseudomonas, Bacillus, Penicillium, Aspergillus etc., secrete organic acids and lower the pH in their vicinity to bring about dissolution of bound phosphates in soil. Increased yields of wheat and potato were demonstrated due to inoculation of peat based cultures of Bacillus polymixa and Pseudomonas striata. Arbuscular mycorrhizal fungi, that is AMF. The transfer of nutrients, mainly phosphorus and also zinc and sulfur from the soil milieu to the cells of the root cortex is mediated by intracellular obligate fungal endosymbiotants of the genera Glomus or Gigospora etc., commonly known as AM or arbuscular mycorrhiza. They possess vesicles for storage of nutrients and arbuscles for funneling these nutrients into the root system. By far, the commonest genus appears to be Glomus, which has several species distributed in soil. Silicate solubilizing bacteria or SSB. Microorganisms such as Bacillus species are capable of degrading silicates and aluminum silicates. During the metabolism of microbes, several organic acids are produced and these have a dual role in silicate weathering. They supply hydrogen ions in the medium and promote hydrolysis, and the organic acids like citric acid, oxalic acid, keto acids and hydroxycarbolic acids, which form complexes with cations, promote their removal and retention in the medium in a dissolved state. Plant growth promoting rhizobacteria or PGPR. The group of bacteria that colonize roots or rhizosphere soil and beneficial to crops are referred to as plant growth promoting rhizobacteria PGPR. The PGPR inoculants currently commercialized that seemed to promote growth through at least one mechanisms, that is suppression of plant disease termed bioprotectants, or improved nutrient acquisition termed biofertilizers, or phytohormone production termed as biostimulants. Species of Pseudomonas and Bacillus can produce as yet not well characterized phytohormones or growth regulators that cause crops to have greater amounts of fine roots, which have the effect of increasing the absorptive surface of plant roots for uptake of water and nutrients. These PGPR are referred to as biostimulants, and the phytohormones they produce include indole acetic acid, cytokinins, gibberellins, and inhibitors of ethylene production. Recent advances in molecular techniques also are encouraging in that tools are becoming available to determine the mechanism by which crop performance is improved using PGPR and track survival and activity of PGPR organisms in soil and roots. The science of PGPR is at the stage where genetically modified PGPR can be produced. PGPR with antibiotic, phytohormone and siderophore production can be made. In biofertilizer production industry, equipments are the major infrastructure which involves 70% of capital investment. Any compromise on the usage of the following mentioned equipments may lead to decline in the quality of biofertilizer. The correct use of equipments will give uninterrupted introduction with quality inoculum. Essential equipments. Autoclave. It is an apparatus in which materials are sterilized by air-free saturated steam under pressure at a temperature above 100 degrees centigrade. If the steam pressure inside the autoclave is increased to 15 psi, the temperature will rise to 121 degrees centigrade, that is sufficient to destroy all vegetative cells. Normally, all growth medium are sterilized in the autoclave. Laminar airflow chamber. Laminar airflow chamber provides a uniform flow of filtered air. This continuous flow of air will prevent settling of particles in the work area. Airborne contamination is avoided in this chamber. Culture transfers and inoculations can be done here. BOD incubators. BOD incubators provide controlled conditions of light, temperature and humidity etc., which is required for the growth and development of microorganisms. Multiplication of starter culture can be done in this instrument. Rotary shaker. It is used to agitate culture flask by circular motion under variable speed control. Shaking provides aeration for growth of cultures. Shakers holding up to 20 to 50 flasks are generally used. The capacity of the shaker may be increased if it is a double decker type. Hot air oven. Hot air oven is meant for sterilizing all glassware materials. Dry heat is used in this apparatus to sterilize the materials. Normally, 180 degrees centigrade is used for 2 hours for sterilizing glassware. pH meter. It is an instrument for measuring pH of the solution using a 0 to 14 scale, in which 7 represents neutral points, less than 7 is acidity and more than 7 is alkalinity. It is useful in adjusting the pH of the growth medium. Refrigerator. This equipment is used for preserving all mother cultures used for biofertilizer production. The mother culture is periodically subcultured and stored in the refrigerator for long-term usage. Fermenter. A fermenter is the equipment which provides the proper environment for the growth of a desired organism. It is generally a large vessel in which the microorganism can be kept at the required temperature, pH, dissolved oxygen concentration and substrate concentration. Different models of fermenters are available depending on the necessity. A simple version model contains steam generator, sterilization process devices and agitator. A sophisticated fermenter contains pH regulator, oxygen level regulator, anti-foam device, temperature controller, etc. Biofertilizers are carrier-based preparations containing efficient strain of nitrogen fixing or phosphate solubilizing microorganisms. Biofertilizers are formulated usually as carrier-based inoculants. The organic carrier materials are more effective for the preparation of bacterial inoculants. The solid inoculants carry more number of bacterial cells and support the survival of cells for longer periods of time. The mass production of carrier-based bacterial biofertilizers involves 3 stages. First, culturing of microorganisms. Second, processing of carrier material. And third, mixing the carrier and broth culture and packaging. Culturing of microorganisms. Although many bacteria can be used beneficially as a biofertilizer, the technique of mass production is standardized for Rhizobium, Azospirillum, Azotobacter and Phosphobacteria. Specific media and inoculum preparation protocols are available for different microorganisms. Processing of carrier material. The use of ideal carrier material is necessary in the production of good quality biofertilizer. Peat soils, lignite, vermiculite, charcoal, pressed mud, farmyard manure and soil mixtures can be used as carrier materials. The neutralized peat soil or lignite can be found to be better carrier materials for biofertilizer production. The carrier material should be cheap, locally available, non-toxic and have water holding capacity of more than 50% and should have high content of organic matter. Peat, vermiculite, lignite, etc. are the commonly used carrier materials. Mixing the carrier and the broth culture and packaging. Inoculants packets are prepared by mixing the broth culture obtained from fermenter with sterile carrier material in a polythene bag. The biofertilizer packet is stored at a cool place away from heat and sunlight. Factors which affect survival, efficiency and performance of biofertilizers. These factors are host plant, soil fertility, cropping practices and then there are certain biological and environmental factors. So far we have talked about the benefits of using biofertilizers, but there are certain constraints posed while using them. Constraints in biofertilizer technology. Though the biofertilizer technology is a low cost, eco-friendly technology, several constraints limit the application or implementation of this technology. The constraints may be environmental, technological, infrastructural, financial, human resources, unawareness, quality or marketing, etc. The different constraints in one way or the other affect the technique at production or marketing or usage level. Technological constraints. It includes use of improper or less efficient strains for production, lack of qualified technical personnel in production units, unavailability of good quality carrier material or use of different carrier materials by different producers without knowing the quality of the materials, and production of poor quality inoculants without understanding the basic microbiological techniques, and sometimes short shelf life of inoculants. Infrastructural constraints. It includes non-availability of suitable facilities for production, lack of essential equipments, power supply, etc., lack of space availability for laboratory, production and storage, etc. Also, the lack of facility for cold storage of inoculant packets. Environmental constraints. It includes seasonal demand for biofertilizers, simultaneous cropping operations and short span of sowing and planting in a particular locality, soil characteristics like salinity, acidity, drought, waterlogging, etc. Constraints due to unawareness on the technology. Unawareness on the benefits of the technology, problem in the adoption of the technology by the farmers due to different methods of inoculation, no visual difference in the crop growth immediately as that of inorganic fertilizers, and unawareness on the damages caused on the ecosystem by continuous application of inorganic fertilizers. Thus, in this module, we have discussed biofertilizers, their importance and various microorganisms used as biofertilizers. In the next module, we will talk about biopesticides.